Hello everyone, and thank you for attending today's webinar, Harnessing Bioanalytical Data in Early Phase Clinical Pharmacology Studies. I'm Sharon Saltz, editor of NeuroCentral at Future Science Group, and I'll be your host for today's event. Before I introduce you to our speaker, I'd like to quickly cover a few housekeeping items. Firstly, all the widgets are resizable and movable, so feel free to move them around to get the most out of your desktop space. You can expand your slide area or maximize it to full screen by clicking on the arrows in the top right-hand corner. Please let us know your thoughts on today's webinar by tweeting us at Bioanalysis Zone using the hashtag BZWebinars, or simply share today's webinar with your friends and colleagues using the Share This widget on your screen. Some networks cause the slides to advance more slowly than others, so logging off your VPN is recommended. If your slides are behind, pushing F5 on your keyboard will refresh the page. You can find additional answers to some common technical questions located in the Help widget at the bottom of your screen. An on-demand version of today's webinar will be available approximately 1 hour after the webinar and can be accessed using the same audience link that was sent to you earlier. So we are delighted to have Dr. Michael Kong from Altasciences presenting for us today. Michael Kong joined Altasciences in January 2017 as Director, Medical Science Liaison. In this role, Dr. Kong helps sponsors navigate through the design and conduct of clinical pharmacology studies. He holds a PhD in pharmacology from the University of Toronto in Canada. We invite the audience to submit any questions you may have for our presenter throughout the webinar using the Q&A widget to the right-hand side of your screen. We will pose your questions to Michael in a live Q&A session after the presentation and then follow up any outstanding questions offline. So thank you again for joining us today, everybody. I'll now hand over to Michael to start today's presentation.
Well, good morning, and thank you, Sharon, for the introduction. So in today's webinar, I'd like to talk a little bit about the types of clinical pharmacology studies that are performed by sponsors and CROs like Altasciences to support early drug development. The scope of my presentation is really to help bioanalytical scientists understand how the information they generate coming out of the lab is used in characterizing drug pharmacology. I think with all the work involved in assay development and validation, the bioanalytical teams may not have the opportunity to get this kind of visibility outside the lab, and hopefully this presentation will be a useful introduction. So just a little information for you about Altasciences. We are a mid-sized full-service contract research organization with over 25 years experience in early drug development. We have 2 clinical sites in North America, one in the US in Overland Park, Kansas, and the other in Montreal, Canada. We also opened a preclinical testing facility in Everett, Washington last summer to help us achieve our goal of providing an integrated research service solution from lead candidate selection to proof of concept. And given the audience that is here today, I should also mention that we have a 25,000 square foot bioanalytical facility about 20 minutes outside Montreal that supports both small and large molecule analysis. So here's an overview of what I will be discussing today. I'll first give a primer on what clinical pharmacology studies are and what they tell us. I'll then talk about some of the key bioanalytical objectives in these studies, some of the applications they're used for, and then some issues that clinical pharmacologists see with some of the data that comes out of the lab. I'll then close with some concluding remarks. So what do clinical pharmacology studies tell us? Well, clin pharm studies broadly address one of 2 objectives. The first being to understand the time course of drug exposure after dosing. This is known as pharmacokinetics and is often described by 4 overlapping processes the body has on the drug: absorption, distribution, metabolism, and excretion. The red curve on the left is from a study subject showing drug concentration levels from plasma over time. If we assume, say, a highly soluble drug is dosed orally at time 0, it enters the stomach, the GI tract, and will be absorbed into the systemic circulation, which is represented by this rise in drug levels to this peak. The decline in drug levels is described by drug distribution into tissues and by metabolic and elimination processes, which are mediated primarily by the liver and kidney. Now, 2 of the key parameters used to describe overall systemic exposure or bioavailability of a drug are the Cmax, which is the maximal drug concentration, and the AUC, the area under the concentration time curve, which describes the extent of drug absorption. The second objective of clin pharm studies is to understand the biological effects or the pharmacodynamic properties of the drug. So these effects are dependent on biochemical and molecular interactions, for example, by interaction with various cellular structures or by binding to the receptor at the site of action. The blue curve on the right is a dose response or dose effect curve, which describes the biological effect of a drug at increasing drug concentration levels. The Emax is the maximal effect of some biological response to the drug, and the EC50 is the drug concentration at which half of the maximum effect is achieved. Now, both of these parameters tell us information about the potency and efficacy of a drug. So here's a list of some of the common clinical pharma studies that would be seen in a standard drug development program. I describe these as early phase studies, which I define as before phase 2B, but in some cases these studies can be done later, even after phase 3. The main objective across all these studies is to answer three main questions. The first one is the drug safe? Now, this will be answered very early on in the first-in-human study, but also in drug interaction studies and TQT studies where cardiac safety is thoroughly evaluated. Second is what doses should be used. So how large a dose will maximize efficacy with minimal toxic effects? What is the frequency of dosing to maintain effectiveness? Should it be dosed once a day, once a week? This requires information on pharmacokinetics of the drug from the first-in-human, comparative bioavailability, and ADME or mass balance studies. And the third is there evidence of efficacy? Are there validated pharmacodynamic endpoints that can quickly tell us that the drug is hitting its target and doing what it's supposed to be doing? Now, for this presentation, I will be focusing mainly on the highlighted studies and discussing the role that bioanalytical data has early in development. When we characterize drug pharmacokinetics, we use drug concentration data to derive information on bioavailability of both the parent drug and metabolites, particularly active metabolites that may have been identified earlier in development using in vitro methods like liver microsomes and in vivo methods like mass balance studies. Clinical pharmacology studies can be used in looking at pharmacokinetic differences in formulations, for example, tablet versus oral liquid, or differences in the route of administration. The choice of which biological matrix is used for analysis can depend on the nature of the drug. For instance, using whole blood instead of plasma to analyze the antimalarial drug Malarone may be more appropriate since it binds readily to red blood cells. In terms of pharmacodynamic objectives, biomarkers are often used in clin pharm studies as a quantitative means of monitoring a biological or pathogenic process in response to a therapeutic intervention. They can be used to inform clinical studies in different ways, for instance, as a screening tool for subject recruitment or as a surrogate for a clinical endpoint. The important thing is the bioanalytical assays need to be fit for purpose, as sensitivity of one validated method may be appropriate for one application but not another. One pharmacodynamic consideration that should be made for large molecule biologically derived drugs such as antibodies is the potential for an immune response to the drug known as immunogenicity. Now, the production of antibodies against a drug of interest can affect the drug's pharmacokinetics or can neutralize the drug's activity, thereby decreasing its efficacy. Usually a three-step process is often used by labs, including ours, in doing this. One is screening samples for anti-drug antibodies using ligand binding assays, and then the reanalysis of these positive samples in a confirmatory assay, and then analyzing these samples for the presence of neutralizing antibodies. So we saw earlier that there are different types of clinical pharm studies designed to meet a number of different objectives, some of which are summarized in the left-hand column of this slide. In order to ensure these objectives are met with speed and efficiency, there are important bioanalytical considerations that should be made. The method should be selective enough to handle potentially interfering substances, such as metabolites or protein degradation products, as well as concomitant medications that we do try to exclude as much as possible in the recruitment of healthy volunteers, but can still be an issue when dealing with disease patients or special populations. The assay throughput should be optimized to rapidly obtain data across a wide dosing range. This is important especially for first-in-human studies, which I will discuss shortly. Pharmacokinetic studies for a single drug are often done in both plasma and urine. For example, if the drug is renally excreted. And so the evaluation of matrix effects in development is important in making accurate measurements to follow the kinetics of the drug through different biological compartments. Lastly, the extent of validation required should be considered in the context of the assay's purpose. For example, if an assay is intended to explore differences in response between formulations, a qualified assay is adequate. If it is intended to show differences in a specific population to support a labeling claim, then of course it should be fully validated. The first-in-human study is often seen as a study that bridges preclinical development to clinical development, as it uses preclinical data to help establish dosing and safety in humans, which is the foundation for all subsequent clinical development. The key objective is to establish the pharmacokinetics, but more importantly, a safe dosing range in normal healthy subjects. A protocol written for a first-in-human study will involve a dose escalation design where different cohorts of subjects will be administered a single dose of the drug. This is called the single ascending dose phase. A cohort of 6 to 8 subjects will be typically randomized to a drug or placebo and evaluated for adverse events by a safety monitoring committee before escalating to the next dose level in the next cohort. In this diagram, there are 6 single dose levels, which is typical for single ascending dose studies. Within the same protocol, a similar dose escalation design, but with multiple doses, can be planned after the SAD phase is completed. Again, this helps to establish safety and assess exposure under steady-state conditions, which is a dosing state where drug concentrations are relatively constant. Now, there are usually a smaller number of dose levels in the multiple ascending dose phase, and dosing is usually conducted once or twice a day. And here in this slide, there are 3 dose levels represented in the MAD phase. Now, more and more sponsors are now combining other objectives into the first-in-human study. For example, studying the effect of food on drug bioavailability, or making cardiac safety assessments, which I haven't listed here. A proof of principle component can also get some preliminary evidence of efficacy in a small cohort of patients, and sometimes this is also integrated into these studies. And so the primary objective is to demonstrate safety after single and multiple doses, and some of the key endpoints in first-in-human studies are the maximum tolerated dose and a summary of adverse events. Pharmacokinetics are often included as secondary objectives with AUC, Cmax, and half-life as key parameters of interest. These parameters can be statistically evaluated for dose proportionality to determine whether dose increases lead to corresponding and predictable increases in exposure. Now, one of the most obvious areas of risk in first-in-human studies is starting dose, more formally called the maximum recommended starting dose. And is the reason why we often recruit healthy volunteers in these studies, as there is a larger safety margin with this population. From the preclinical safety data in animals, we use an algorithmic approach to define the MRSD, which begins by obtaining the no observable adverse effect level, which is the highest dose tested in an animal species that does not produce any significant adverse events. Using this dose, we apply a conversion factor to obtain a human equivalent dose. Now, the FDA has published a table of conversion factors for various animal species, which I've shown here on this slide, and it helps drug developers do their allometric scaling. Often, you'll have safety data from a rodent species and a non-rodent species, for example, dog or monkey, and we usually use the lowest human equivalent dose from the animals tested, which is often the most sensitive species evaluated. We then apply a safety factor to the human equivalent dose, which is typically done by dividing by 10 to account for physiological differences and uncertainties between animals and humans. In the end, final dose adjustments might be made. An increase in the safety factor may be justified if the drug is a new class of drug or shows toxicities that are not easily detected. Conversely, it could be decreased if it's from a well-characterized drug class or if toxicities are easily monitored and managed. Now, there are other methods of MRS determination, for example, the minimal anticipated biological effect level and the minimum effective dose, but these are beyond the scope of this presentation, and I'd refer you to some of the FDA and EMA guidance on this topic if interested. So even after we've established what we think is a safe starting dose, In dosing the first cohort of subjects in a first-in-human study, CROs and sponsors will often use a sentinel dosing strategy whereby 2 subjects are randomized 1-to-1 to receive either active drug or placebo. And this is because there's still a level of unpredictability that can't always be accounted for with new drugs, especially those that may be from a novel drug class. And once an adequate amount of time has elapsed to monitor for safety in these 2 sentinel subjects, the rest of the cohort is randomized to the remaining treatments. Now, sentinel dosing is typically, but not always, done at each dose level, and this helps to ensure minimum human exposure to a drug that may have unforeseen AEs, for example, due to nonlinear pharmacokinetics. And the Safety Monitoring Committee will look for protocol-defined stopping or, in more severe cases, study stopping rules. And the PK data from the lab is often an important part of these stopping rules, and the committee will want to see whether the systemic exposure of the drug aligns with emerging safety data and if dose escalation is justified. Dose escalation tends to move as quickly as prudent monitoring of safety and will often require PK parameter data within 48 hours of dosing. So it's ideal to have an optimized method with a quick turnaround time to support safety meetings for decision making. Thoroughly defined PK profile is not usually required, but estimates of the area under the curve and Cmax are typically the minimum parameters needed. Also, if a drug doesn't exhibit linear pharmacokinetics, particularly at higher dose levels, where there may be capacity-limited metabolism, expect your Cmax values to be outside your expected range, so dilutions at this point may be necessary. Now, as I mentioned earlier, a separate cohort of patients is sometimes integrated with a first-in-human study to establish early proof of efficacy. And biomarkers can be utilized as pharmacodynamic endpoints as a means to study this, the selection of which depends on data gathered from preclinical or in vitro studies. Post-dose measurements at a time expected to yield a pharmacological effect are usually compared to pre-dose measurements and reported as a change from baseline. Now, typically at this early stage, biomarkers are exploratory in nature, intended only to make go-no-go decision-making, and so full validation is usually not done. If used in Phase 2A study, however, as an endpoint secondary to a primary clinical endpoint, full validation would be required, and so we come back to this notion of ensuring the assays are fit for purpose. In patient studies, the use of biomarkers can be used to diagnose a condition that may be an inclusionary requirement for a study. For example, at Altasciences, we recently used a panel of non-invasive biomarkers with specific cutoff points to identify patients that were likely to have non-alcoholic steatohepatitis. This had advantages over histological diagnosis through liver biopsy in that it helped facilitate rapid screening and recruitment. Now, comparative bioavailability studies play an important role in the development of this first-in-human formulation. Once safety is established, sponsors may want to modify the formulation to have certain desired properties. They may want that it have extended release characteristics to prolong systemic exposure, or they may want to increase the solubility of the drug so that it is more efficiently absorbed. In assessing PK, again, Cmax and AUC, more specifically AUCT and AUC infinity, are the key parameters of interest, and I'll discuss shortly why an accurate analytical range becomes important in a bioavailability assessment. In generic drug development, this comparison of Cmax and AUC is statistically powered in a bioequivalence study to formally show that it has similar pharmacokinetics to a reference or branded formulation. It is a key objective in the marketing authorization of a generic product. In new drug development, however, bioequivalence studies per se are not mandated from a regulatory point of view, but still may be done by sponsors to scientifically establish equivalence for some of the reasons I mentioned earlier. They are often done in post-approval scenarios where a manufacturing site has changed or an excipient has been added to the formulation, for example. Now, comparative BA studies are usually conducted as crossover designs where the same subject is randomly given the test and reference formulation in 2 different time periods. Now, this is done to reduce the between-subject PK variability that is more evident in parallel design studies. So it makes it more likely to statistically show bioequivalence when 2 formulations truly are bioequivalent. Now, drug-drug interaction studies are a type of PK study where the investigational drug might be assessed for its ability to inhibit, induce, or be a substrate of certain metabolic enzymes or transporters. Data from preclinical and in vitro studies will guide the specific metabolic enzymes or transporters to be evaluated. Major drug interactions can reduce the efficacy or increase toxicity, which has led to market withdrawal of some drugs. If you remember, the popular antihistamine Seldane was withdrawn from the U.S. market for cardiovascular risks associated with drug interactions. Now, in this example, drug TG12, which was shown to be a strong substrate of the metabolic enzyme CYP3A4 from human liver microsome studies. Itraconazole, it's a well-known inhibitor of the CYP3A4 enzyme, was used in this study to evaluate its drug interaction potential. Now, the study was essentially divided into 3 parts. Part 1 is when subjects receive a single dose of TG12 on day 1 and PK sampling for 3 days. Part 2 involves dosing itraconazole on day 4 and then daily to day 9 to maximize CYP3A4 inhibition. And then Part 3 is where TG12 is dosed concomitantly with itraconazole on day 10, followed by PK sampling again for 3 days. And so when you compare the 2 TG12 PK profiles with and without itraconazole, which is the green and red graphs respectively, you can see that there is an almost 70% increase in the bioavailability of TG12 when CYP3A4 is inhibited. Now, safety assessments that are also done in DDI studies can also inform of any evidence of toxicity and ultimately precautions that should be taken in patients using 3A4 inhibitors with this drug. Let's now discuss some issues that clinical pharmacologists see with data that often require collaborative investigation to avoid reporting erroneous results. In pharmacokinetic studies, it is common for the bioanalytical scientists and the clinical pharmacologists to work closely together prior to sample analysis to define data transfer specifications or the appropriate assay validation range for a drug. Now, when sample analysis is complete, the clinical pharmacologist constructs pharmacokinetic profiles and examines the bioanalytical data prior to calculating PK parameters. Now, the example shown here is a case of inadvertent sample switching in the lab, which can occur simply through human error as samples are being processed. It is sometimes detectable if the switch occurred within the same subject's samples, like in this example, where it appears the 8-hour sample was mixed up between the test and reference samples. Sample switching becomes much more difficult, however, to detect and defend if it occurs between subjects. Now, in this example, we have an issue where the 8-hour sample is below the limit of quantification, which in itself is normal, but because it falls between 2 samples that are well above the limit of quantification, would appear suspicious. Physiologically, drug levels just do not disappear and then reappear in the blood with such a large change in magnitude. So, although this could be due to a sample switch, sometimes the cause is simply not known, and we need to rely on predefined data handling rules to get around this anomaly. Now, this example is a case where pre-dose drug levels are observed for the reference formulation, which is the red curve. You can see the time 0 sample has a measurable value which would be expected to be 0 since the subject would not have been dosed at this point. For non-endogenous synthetic drugs, this is usually a sign that something wrong occurred in the clinic. For example, the subject was already using the tested drug prior to dosing, or there was sample contamination, which actually can happen with inhaled products if the drug aerosol from another subject dosing is not properly cleared from the air. Now, the FDA recommends that subjects with pre-dose levels greater than 5% of Cmax be removed from the statistical analysis of the PK data. One of the key PK parameters that the FDA suggests in comparing drug bioavailability is AUC infinity. AUC infinity is a theoretical measure of the total systemic exposure of a drug until it is completely eliminated from the body. Now, as you can see from the formula on the right, it is calculated by combining the measured area under the curve to the last measurable time point, called AUC 0 to t, with the extrapolated area under the curve calculated as the last measurable concentration divided by Kel, the elimination rate constant. Now, Kel can be mathematically derived, but it can also be empirically derived from the drug concentration time profile. Now, if we convert the concentration axis on the left to the log scale in the graph on the right, you can see that the terminal part of the curve exhibits a mono-exponential decline. And I've circled this in green. Without going too much into theoretical pharmacokinetics, Kel can be derived from the slope of the last 3 points in this terminal phase. Now, a problem that can be countered is if your lower limit of quantification is not low enough. You may not be able to measure the terminal part of the curve, which will not allow you to derive Kel and ultimately AUC infinity. Now, it becomes important here to work with your clinical pharmacologist, especially in pivotal bioequivalence trials, to define an LLOQ that is sensitive enough to capture the elimination profile of the drug. In general, sample reanalysis for pharmacokinetic reasons is a sticky topic that, if done, should be guided by an objective decision-making process. And this is because reanalyzing samples can be perceived as a way to obtain more favorable data to arrive at desired outcomes. Now, the important thing to note is that any decision taken should be guided by standard operating procedures that clearly justify the course of action. The last topic I'd like to touch on are clinical pharmacology studies with special populations that may use the drug, but may require dose adjustments due to the nature of their disease. Now, recall we mentioned that one of the objectives in clinical pharm studies is defining appropriate doses to use in different populations. Patients with renal impairment are likely to have pharmacokinetic differences if the drug is known to be cleared primarily through the kidneys. Therefore, a drug that is likely to be used in these patients, for example, in the elderly, will require a study comparing the pharmacokinetics in chronic kidney disease patients with healthy subjects. Patients with varying degrees of renal impairment are recruited, and biomarkers are often used to define severity and therefore study eligibility. Serum creatinine is a common biomarker that's used in renal impairment studies. That's measured and used in one of a number of different equations to assess kidney function. Cockcroft-Gault and the modification of diet in renal disease are two of the more common equations that are used. Similar studies in hepatic impairment patients also need to be done for drugs cleared by the liver. In order to define severity of the disease, we usually classify patients using the Child-Pugh score, which is a composite score of 5 clinical measures of liver disease. 3 of these measures use the lab biomarkers albumin, bilirubin, and ascites. Now, the pediatric and elderly are not necessarily a disease population, but do have pharmacokinetic differences that need to be studied. One of the challenges faced with this group is that PK sampling and the blood volume limitations. Fortunately, there are alternative blood sampling methods out there that focus on microsampling technologies that permit microliter amounts of blood to be analyzed. These also allow subjects to sample at home without the need to return to the clinic. And so I've listed a couple of microsampling techniques that we currently use for studies at Altasciences in these populations. So based on everything I've discussed, I would like to conclude with just 2 take-home messages. The first is that early clinical development is a bioanalytically driven process that heavily relies on data that is key in interpreting pharmacokinetic and pharmacodynamic questions about new therapeutics. The second is that a collaborative effort between the lab and the clinical pharmacologist is required in an objective setting and feasibility assessments, and together will help establish expectations earlier in clinical development. And with that, I thank you for your time and attention, and we'd be happy to take any questions.
Thank you for that very insightful presentation. I hope our audience found it of great interest and value. If you do have any questions for Michael, then please continue to submit them using the Q&A tool on the right-hand side of your screen. So we have received a number of questions so far, and I will address some of these now live on air. Our first question is, with regard to using biomarker assays for study eligibility, when is it appropriate to use local versus central labs in multi-site studies?
Right, so I think whenever it's possible, I think central labs should be used for eligibility since different clinical sites will have different validation standards, different instruments, different sample collection requirements. One scenario where I would say central labs is absolutely required is when your marker is being used to support an efficacy endpoint. So just to give you an example, in oncology, sometimes you would screen patients that harbor certain mutations, and this can greatly influence therapeutic outcome. So you want to ensure that you have these patients that have these specific mutations. So it's important both from a scientific and a regulatory perspective that these sort of endpoints be standardized as much as possible when it comes to efficacy. That being said, I think it's still acceptable to use local labs if there exists a clearly validated diagnostic test. So for example, an off-the-shelf assay where you can be more confident that it will give you consistent results.
That's great. And our second question is, in first-in-human studies, you talked about the single and multiple ascending dose phases being conducted in the same protocol. So our lab occasionally receives samples from both phases in parallel. Can you explain why these studies are sometimes conducted together?
Yeah, that's a great question. So just to step back a bit, so traditionally, single ascending dose, multiple ascending dose studies, they are run as separate studies, typically in sequence with the SAD study run first, followed by the MAD. But now, more and more sponsors are conducting both the SAD and MAD studies in what's called umbrella protocols. So they're done really as one single protocol in which both the single and multiple dosing objectives are achieved almost at the same time. And so normally what you would do is data from the first few SAD cohorts, once they become available, your clinical pharmacologist can do software simulations to predict what the pharmacokinetics of the first multiple dosing cohort would look like. And if they conclude that it's predicted to be safe, or if the achievement of safe systemic levels is predicted, then the dosing of the first MAD cohort can begin as early as after safety has been established for, say, the third SAD cohort. So really you're doing both in parallel, and so you sort of trim your timelines down by almost 30% by doing this, which is one of the main reasons sponsors and CROs look to do this as an option to the sequential approach instead.
Okay, that's great. And our third question is, in your DDI example, the drug was a substrate of a single CYP. Most drugs are substrates of multiple CYPs. Do you have to run a DDI for each type?
Yeah, so the example that I gave in my slide deck was evaluating which CYP enzymes metabolize the drug of interest. And so you can use a CYP inhibitor or inducer to see how levels of your drug change when dosed concomitantly. So typically these kind of studies would be done as individual studies for each CYP. But I also mentioned that DDI studies are also used to evaluate drugs as CYP inhibitors or inducers, in which you would use known probe substrates specific for certain CYPs. So in these type of studies, you can dose your drug as essentially cocktails of these probe substrates, which will allow you to simultaneously evaluate which CYPs your drug affects in vivo. Now, it's important that if you do this, you use substrates that are specific to a single or individual CYP. And FDA has published a list of suggested probe substrates in their DDI guidance document, so you can refer to that for more information.
Brilliant. Thank you very much, Michael, for answering our audience questions today. Before we wrap up, do you have any last comments you would like to add?
No, just to say thank you for your attention, and if you do have any other further questions, feel free to reach out to me. I think my contact information is going to be made available shortly.
Okay, that's great. I would like to once again thank our wonderful presenter, Michael, as well as you, our audience listeners, for your time and your questions. If you would like to let us know your thoughts on today's webinar, then please feel free to complete our feedback survey in the resource widget or tweet us at Bioanalysis Zone. Don't forget to also visit us at www.bioanalysis-zone.com for more webinars. Thank you again for attending. And